Hello, this is Dr. Paul Cottrell, and I'm going to talk about Mpox today. The scientific side of this. None of the videos that I have seen on YouTube dealing with Mpox have gone into the detail that I will be going into. So I think this will be a good learning lesson. It's, at a kind of a graduate level, so it's important to kind of pay attention to what I have to say. A lot of the stuff that the mainstream media is saying is just very sugar coated and it's not giving you the details. So I think it's very important that you review this video multiple times so you understand what I am talking about when it comes to Mpox. But before we do that, can you please go to my store, the-studio-reykjavik.com, and help support my work by donating. You can donate on the homepage at the very bottom. You can donate through Stripe, PayPal, or buy me a coffee. Just click the buttons there and it'll take you to the donation page and donate that way. In addition, I have lots of different products. I have ebooks and different supplements, but I want to focus on some new supplements that I have. I have the olive leaf extract. This will help to boost up your immune system and help with your cardiovascular system. So you should be taking this every day. The olive leaf extract helps with the immune system by bringing down inflammation, and it helps with the cardiovascular system by improving your HDL levels. It increases your HDL levels and it lowers your LDL levels. So please go to the store and get the olive leaf extract to improve your immune system and your cardiovascular system. I also have zeolite. Zeolite is a detoxifier, a heavy metal detoxifier. It also detoxifies chemicals and toxins from your bloodstream. How it works is that zeolite is a molecule that is negatively charged. So it's going to attach to positively charged heavy metals, toxins, and chemicals to help detoxify. So take this every day, and it will soak up those toxins and heavy chemicals. If you're on medication, you should talk to your doctor first before using zeolite, because some of the medication that you might be taking, zeolite might take out of your bloodstream. So just keep that in mind. But for the ones that aren't on medication, it's really important to detoxify your body on a regular basis. Some people detoxify their body every day. Some people do it once or twice a year. It's up to you. But this type of zeolite you can take every day. So please go to the store, the-studio-reykjavik.com, and get the zeolite. The link is in the description of this video. I also have a new product in the structural nanosilver section, and that new product is the cherry silver soothing lozenge drops in 100 count. So basically, it's a new flavor that we have here. It's the cherry flavor now. It's in 100 count. This is made from structural nanosilver. It'll help to neutralize pathogens and boost up your immune system. It will also improve the mucosal barrier in your oral cavity. So by doing that, it will reduce the chances of getting infections. In addition, I have a lot of new products underneath the studio products. It's 5 new products that I have brought onto the store. These are supplements that I've been taking every day for quite a while, and now I'm able to provide them to you, and you add it to the stack that I'm doing, and you'll notice that you're slowing down the aging process, you're boosting up your immune system, and you are improving your longevity. So the first one is NAC. NAC is really important because it brings up your glutathione levels. It's a great antioxidant. It's synergistic with the other antioxidants that I have on my store. So you should be taking NAC every day. It also helps with detoxifying your body. So do that. Take NAC every day for detoxification and as an antioxidant by boosting up the glutathione levels. and it's synergistic. NAC is synergistic with C60. Ubiquinol, which is the active form of CoQ10. This is really important to be taking every day. Ubiquinol is a compound that will help to boost up your energy levels. It's an antioxidant, and it'll also help to protect your neural health. So please take the ubiquinol that I have on my store. I take it every day. So again, it's also synergistic with the other antioxidants that I have on my store. NAD plus, very important to boost up your energy levels. It helps with also your brain function, your neural health. So please go to the store, the-studio-reykjavik.com, and get the NAD plus. It also helps to improve repair of your mitochondria and your DNA. So that's really important, to maintain good cellular health, but also your ATP levels. So you should be taking NAD plus every day. It is synergistic with C60 and resveratrol and niacin. Vitamin K2, MK7. This will help with your bone health and your cardiovascular health. So you should be taking that every day. I have mentioned on other videos how to stack it to reduce cardiovascular disease, how to reduce plaque, and how to remodel your bones and your joints to reduce osteoarthritis. So please go to the store and get the K2 for improving your bone health and your cardiovascular health. Nattokinase, really important. Should be taking it every day. At higher doses, it will bring down LDL levels and bring up your HDL levels, bring down your cholesterol levels. Really important. In addition, it is fibrinolytic, so it will help to break up clots. Should be taking it every day. And it is synergistic with the digestive enzyme complex that I have on my store. So please go to the store, get the new products that I have, the NAC, the ubiquinol, the NAD+, the K2, and the nattokinase. Please go to my store, the-studio-reykjavik.com. The link is in the description of this video. So what I'm going to do is now we're going to start the presentation that I built up on Mpox. Okay, so what we're going to do here is talk about Mpox, and this is a detailed overview. Now, it's important to understand that there are these viruses called pox viruses. And we have like three main categories within the pox viruses. We have smallpox. We have something called the molluscum contagiosum type viruses and the zoonoses viruses. And the last category includes the Mpox. Now, the second category, the molluscum contagiosum, is primarily in the keratinocytes. So it depends on what cells get infected and all this stuff. But this presentation is going to be focused on Mpox. The structure of a pox virus, all right, is much more complex than, let's say, your other viruses that we've been talking about in the last 4 years. This virus is much more complex, and it has an envelope. All right. They have lateral bodies. They also have a membrane, a core membrane, and a core. And underneath the envelope is also an outer membrane. So you have an envelope, an outer membrane, lateral bodies, a core membrane, and a core. And inside the core are viral DNA and proteins. This is where all that really important machinery is coded. All right. And the proteins, the important proteins that are needed. This is a very complex virus. It's not fully understood. And on the surface of the protein are these tubules, all right, these surface tubules. Now, with an electron microscope, you can see the actual pox virus, all right, and you can see the envelope and the outer membrane and the core, all right. Now, pox viruses are different. Pox viruses are 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 different from other DNA viruses. Now, how or why is that? It's because their whole entire replication cycle is in the cell cytoplasm. It's not using the DNA, it's not using the nucleus of the cell that it's infecting. So a pox virus goes in to a cell and it will replicate in the cytoplasm. It will not need the cell nucleus. And pox viruses are DNA viruses, all right? So just keep that in mind. And the pox virus has a very large genome. It has anywhere from 100 to 200,000 bases in its genome, and it codes for about 191 genes. So the pox virus codes for enzymes that will help with the production of the messenger RNA and its own DNA production within the cell cytoplasm. So it doesn't need to hijack too much of the cell. It just needs basically the cytoplasm. All right, so it's a complex virus. Pox viruses are, in a way, almost like a mini cell, all right? But it is considered a virus, and it needs the cytoplasm for replication. So here is an example of how the virus works in the cytoplasm. So you have a cell, all right, and you have the nucleus, and the nucleus isn't involved. So the pox virus will attach, and we'll go into detail about the attachment and the fusion, but it attaches and fuses, and what happens is it loses its membrane. And that membrane is, when it loses its membrane at the loss of the outer membrane, then the core is exposed. And remember, there's a core membrane also. And so there are these things called late transcription and early transcription. So what happens is that during the early phase, the core has DNA and certain types of enzymes, right? And so what happens is that what's going to happen is that you're going to, or certain proteins. What's going to happen is that there's going to be an early transcription. This early transcription is going to lead to early messenger RNA, and those early messenger RNAs that are being formed will create early enzymes. Those early enzymes will then feed into uncoding the core and releasing the viral DNA. All right. And when it's releasing the viral DNA, then it will have these DNA strands, right? And then these early enzymes are also DNA polymerase, which basically helps to replicate the DNA. This takes 2 to 5 hours. And then what happens is that it'll go down the late transcription cycle, where now you have DNA that has been replicated. There's late transcription. So you got now late messenger RNAs, you have late enzymes, and you have late viron proteins, okay? You also have early viron proteins that come together with the replicated DNA. And what happens is now you're going to have a situation where you have what is called a spherical immature particles. Right? So you have a zone where there's immature particles. So you have proteins and you have DNA, replicated DNA that's immature, that's just kind of near each other. But over time, what happens is that there's a morphogenesis where in 4 to 24 hours, the spherical immature particles start to coalesce and start to form virus. And that virus is then released by the cell, by cell disruption, right? Or exocytosis. So the cell may blow up for whatever reason, or just bud off, and there's an exocytosis. Okay, so let's go over this again because this is important. The virus replicates in the cytoplasm only and does not involve the nucleus. It attaches to a couple different types of receptors. We'll go over that. And it fuses based on some complex fusion proteins. There is a loss of the outer membrane, and then the core is exposed. There are early transcription of messenger RNA, okay, that is happening. And those messenger RNAs are then, because there's DNA here, so there's enzymes in here that will start to create messenger RNA, and those early enzymes are then going to start releasing the DNA for it to be replicated. But it also, those enzymes will help to produce the early viron proteins. Now, as the DNA is being released out of the core from an coatase, right, from the early enzymes that were, during the early transcription, this core is being uncoated, the uncoating of the core. The DNA is released, and now there's also DNA polymerase that is part of the early transcription that leads to the replication of the DNA. The DNA, right, is replicated, and some of that DNA that's replicated will end up doing late transcription, and those late transcription will lead to late enzymes and late viron proteins. So the early path creates early viron proteins and really important enzymes for uncoating the core and replicating the DNA. The late enzymes will lead to helping with the late viron proteins. There will be a spherical immature particles that are floating around as a body, and those will be late viron proteins and early viron proteins and replication DNA. These spherical immature particles will then eventually coalesce in 4 to 24 hours, or in 4 to 20 hours, and eventually a pox virus particle will form and be released through exocytosis or the cell disruption. Okay, so that's really important to understand that the core has proteins and DNA, and that the early transcription helps to release the DNA and create early viron proteins. And the release of the DNA, then it's able to replicate through the early DNA polymerase, and then there's late viron proteins that are created, and then they coalesce. So it's a complex process. It doesn't need the nucleus to do these things. So this is important because this shows that through a virus, you can do a lot of stuff in the cytoplasm. Well, what happens if you package something through biotechnology where you have DNA polymerases and RNA polymerases as part of virus-like particles as some sort of biotech? Then you can hijack cells and do certain things. So that's an important point that I think people need to understand. Cell surface receptors. It's not well understood with pox viruses, but the current research is showing that chondroitin sulfate receptors and heparin sulfate receptors are involved. For the heparin sulfate receptors, it will bind by a protein that's on the virus. That protein is called H3. Now, we're going to go into a little bit more, as we said about the binding. So there's binding and fusion. Binding is where the virus is binding to the attachment of the cell, so some sort of receptor. And then there's the fusion, where it's starting to merge. The virus merges with the cell's membrane, and releases its core. So with the viral binding onto the cell surface, there seems to be three proteins that are involved, and that's the H3, the A27, and the D8 proteins from the pox virus, all right, that are on the surface of the virus. The cell fusion is based on 11 conserved proteins. Those 11 conserved proteins are F9, L1, A28, H2, G3, L5, A21, J5, O3, A16, and G9. Now, remember, I have mentioned that the hijacking of the innate immune system, when it's in this phase here, when it's trying to replicate, the cell may try to use its innate immune system. Well, there are proteins that are in the core that either through early transcription or are just in the core that will actually help to shut down the innate immune system. One of those proteins is E3. Okay, so you have these cell fusion conserved proteins, right? There's 11 of them. And they build a fusion protein complex. That fusion protein complex is F9, L1, which is in this configuration. The A28 will fuse with the H2. The G3 will attach itself with the L5. The A21 J5 will be kind of separated, but be near an O3. And the A16 will be attached to the G9. So you have like these three attachments, and then some other one. You have kind of like a fusion between A28 and H2, G3, L5, and A16, G9, and then you have these others that come and attach, which is the F9, the L1, the A21, the J5, and the O3. So just kind of keep that in mind, that there is this complex protein fusion that allows the virus to be able to move into the cell. All right. So this is, again, pox viruses are complex. This is another electron microscope staining. And you can see you have the cell surface right here. All right. This is the cell. And then the pox virus is here. The pox virus is right here. The core. So you have the pox virus membrane. You have the pox virus surface. You have the membrane of the core. And you have the core. All right. And there's this also an outer membrane here. Okay. You can see that the surface, which has the fusion complex of these proteins, starting to merge with the surface of the membrane, that surface membrane of the cell, and the core is now being released into the cytoplasm. Okay, another good picture of this is that here's the virus. Okay, here's the core. This is the outer membrane of the core. I'm sorry, this is the core membrane. I'll call it the core membrane. This is the surface membrane of the virus. Here's the outer membrane of the virus. All right, and here there's a fusion between the surface and the surface of the pox virus merging with the actual cell surface. Okay, now you can see that the core is being released into the cytoplasm. And there's DNA and proteins in here that will lead to early transcription and late transcription. Okay. Pox viruses are extremely complex. Now, the immunopathogenesis of Mpox, really important to go over, and we're going to spend a little bit of time on this. Okay. So the immunopathogenesis. So how's this thing? It happens a few different ways. Okay. There's a respiratory route. Okay. There's a skin route, which is what we've been mostly focused on. There is a draining lymph node route. There are the route through the liver, the route through the sexual route, infective pustules, and infective ulcers in the mouth. Okay, so let's walk through maybe not necessarily in order, but we can yeah, we can do it in order. All right, so respiratory route first. All right, so you have a mucosal lining and you have ciliated cells and you have these different cells in your respiratory tract, right? And you have dendritic cells, which are part of your immune system. Well, the Mpox virus can infect the dendritic cells and your macrophages. So you have dendritic cells and macrophages. So the Mpox gets in, and what happens is that the dendritic cell is infected. And what happens there is that dendritic cell then will move into a lymphatic vessel. Same thing with the macrophage. A macrophage will get infected and will go into the lymphatic vessel, or you can have actually Mpox virus go directly into the lymphatic vessel. Okay, the skin route. The skin route is that okay, there's some sort of disrupted barrier. All right, irritation on the skin or a cut or whatever, and there's a virus that can get beyond that keratin level and get into your dermis. Okay, in the dermis, in the epidermis layer, all right, you're going to have these Langerhans cells, right? It's part of the immune system. The Mpox can infect the Langerhans cells, and the Langerhans cells will get into the lymphatic system. All right. Or you can have direct virus getting in the lymphatic system. Or you can have fibroblasts that are, because in the dermis layer, so you have keratin, you have epidermis, you have the dermis. So in the dermis layer, you're going to have fibroblasts. So you may have a situation where the Mpox is infecting the fibroblasts, the macrophages, or the dendritic cells in the dermis layer, and then those cells then migrate into the lymphatic system. So you have some sort of infection by the fibroblasts, and then the macrophages come in, try to gobble up some of the fibroblasts show some sort of inflammatory response. The macrophages and dendritic cells chemotax over, and they start trying to eat up these viruses, and then they go into the lymphatic system. Now, as it's draining into the lymphatic system, then what happens is that now you have an infected dendritic cell or macrophage. Now remember, from the respiratory route, it's getting into the lymphatic vessel with the dendritic or macrophages. The skin route is also Langerhans, dendritic macrophages are getting into the lymphatic route or direct virus. And then now you're in a lymph node. Okay. So it's coming in to the lymph node. And now you have your dendritics, your macrophages that have been infected, maybe even some viral particle in the lymphatic system directly into the lymph node. And it's now draining into your lymph node, and then it's spreading, okay, spreading through your lymphatic system. Okay, now in the liver, the hepatocyte, you can have virus in there, and then you have the Kupffer cells that come in and try to eat those up, eat the infection, and then that will also get into the lymphatic system. You also have a sexual route where that it's potentially in the semen, and then it gets into the ovaries. You have infective pustules that are on the body. So that's the typical physical manifestation of Mpox. And you have the aerosol, it can be aerosolized out. So now you have the respiratory, it can get into the respiratory system. It can get in through, which is typically the skin route. It gets into the lymphatic system and spreads. It can also be in your liver, your hepatocytes. It can be through the sexual route. All right. It manifests as pustules, and it can spread from the pustules. It can also spread from some sort of droplets. I wouldn't use the word aerosolized. I would just use droplets. It's not airborne. It's more a droplet form. And it's primarily because of these infective ulcers that you have in the mouth, and you can get these droplets from that. So that's basically how it's working with the macrophages and the dendritic cells and the Langerhans cells, the Kupffer cells. Okay, so that's the immunopathogenesis of Mpox. Now, how does it do the immune evasion? Now, I talked about this in a different form where I talk about the E3 protein inhibiting the innate immune system. The innate immune system is turned off because Mpox is hijacking the cell and pretending that pretending that it's not foreign. It's telling the cell, don't go into apoptosis, don't try to kill me. Right? It disguises itself through the E3 protein. This E3 protein shuts down the RIG1 pathway, shuts down the OAS pathway, shuts down the PKR pathway. It shuts down MAD5. So, and it also shuts down the activation of CD4 and CD8 cells. So this is saying it a little differently. Okay. It doesn't talk, this graph here doesn't say that's the E3 protein, but like I said before, the interferon, cellular interferon, is actually inhibited by the E3 protein from the Mpox. So, okay. So what happens is you have a cell that's infected, and it wants to send a signal to surrounding cells that, hey, there's cytokines. Well, the E3 protein shuts down this interferon alpha beta pathway and turns off the innate immune system from going into antiviral mode. It also does this, it reduces cellular activation, and that basically means that it's not activating the CD4s and the CD8s. There are proteins here, and some of them are some of the inhibition of the cytokines and the chemokines are inhibited by E3, but I believe it's not just E3 that targets these cytokines. All right. There's also blocking of the complement cascade to preventing the cell from getting going and getting killed by the immune system. There's also and I talked about this already with the RIG1 pathway, where the E3 is blocking the PRR signaling cascade and preventing the cell from recognizing foreign DNA and foreign messenger RNA. It's stopping the signaling of IRF3, and it's stopping the signaling of the interferon, not the interferon, the stops the signaling of the NF kappa beta pathway. This is pro-inflammatory. This is a major pathway to get the cell to go into apoptosis. If you block this pathway, then what happens is that you are preventing the cell from dying. And so it can't go into antiviral mode. So this pathway is really important for the Mpox. And because of this, it's escaping apoptosis, like I said. E3 is a major, but not the only one, but is a major protein that's doing these. The reducing the cellular activation, I don't believe is the E3 protein. I believe that's a series of other proteins. Now, I do have a chart in the next slide that goes into detail of some of the proteins. I don't have the time to really go into detail on each one of these, but at least there's a reference there for you. On this slide, we want to talk about the immune evasion. So basically, it can block the TNF, it can block INF gamma, IL-1 beta, IL-18, and IL-6. So it can block these pro-inflammatory responses for the immune system. And it's important to understand that there is this clade 1 and clade 2. We talked about clade 1 A and B and clade 2 A and B. Now, what is important here is that the Pox Zaire strain is clade 1. The difference between clade 1 and clade 2 is that clade 1 is from Zaire. Clade 2 is from West Africa. And that clade 1, Zaire, expresses a protein called D14. And this is part of the protein that blocks that complement cascade that we were talking about on this slide here, blocking the activation of complement cascade. So this is the Zaire strain, clade 1. Now, if you don't have D14 expressed, because that gene is non-functioning or whatever the reason why, that is the clade 2. Clade 2 does not express D14. So that's the big difference. And again, Zaire is clade 1 and West Africa is clade 2. And clade 2 and clade 1, big differences are that clade 1 expresses D14 and blocks the complement cascade. So that's why clade 1 is worse than clade 2. Now, there's a clade 1A and a clade 1B. We don't know yet. We don't have a good understanding what the main genomic differences are between clade 1A and clade 1B. It could be an enhanced function of D14. I suspect that it's an enhanced function of something else, but that needs further investigation. These two slides here are a list of the proteins. I'm not going to go into the details of each one, but here you have Central African and West African. Central African is Zaire. West African is clade 2, clade 1, clade 2. All right. And you have protein function, and we have the gene, and we have the size, the number of amino acids. All right. So we can look at a certain gene, right? And you can see what the function is, right? So here, you have a lot of inhibitor for the NF kappa beta activation. So this shuts down the innate immune system. It's shutting down the BCL2 protein. There's also the IRF3 inhibition. There's double-stranded RNA binding protein inhibitor of interferon signaling with this F3L for the Zaire version, so clade 1. But for clade 2, it's using a different protein. It's the OPG065 protein. The size of the protein is the same. All right, so the point here is that there's a little bit of a complex difference. The interferon binding proteins, B16R, and interferon gamma, B9R, for the Zaire clade 1, but different for clade 2. So so the nomenclature is a little different, probably because of location. Apoptosis inhibitors, right? Which is the B12R, but also B19R. And then on the next page, we also have interleukin 1 beta binding protein, which in this case is on clade 1, but not in clade 2. So that might be another reason why clade 1 is worse, while clade 2 doesn't have it. So maybe there's an enhanced function, maybe some sort of enhanced function with the interleukin 1 beta for the genome. Here's the D14, the inhibitor for the complement that we talked about earlier. So it looks like there's 2 genes that are not in clade 2, which are the B14 and the D14. And that B14 is the interleukin 1 beta binding protein. Let's see on the other slide, is there? No, there are analogs. So I didn't want to go into each one of these. I mean, that's like a whole dissertation there, but you get an idea that you can use this as a reference. Now we know the genes, what they do, the genes that are in clade 1 or clade 2, what they do, and by referencing this, we would understand what kind of mutations that may have emerged between 1A and 1B. Where was the enhancement? That would be, further discussion. Let's see here. And then the last slide here that I have is that pox virus production. It can create 10,000 or more viral particles in each infected cell. And because it's shutting down the innate immune system, the adaptive immune system with these different proteins that I talked about, stopping the activation of CD4 positive and CD8 positives, stopping the apoptosis pathway, it can create lots of protein, lots of DNA, and package it in a cell. So if it is flying underneath the immune system, the innate or the adaptive, then it's easy for it to spread on the body and eventually, really take hold and show a major manifestation on the skin. Pox viruses are complex. They have lots of proteins. They have 191 proteins. They have a DNA genome, and it does not need the cell's nucleus. All it needs is the cell's cytoplasm. So with that said, that covers the presentation for Mpox in a detailed overview. Hopefully you learned something. Please help support my work by donating. You can donate on my website, the-studio-reykjavik.com. You can also donate by being a paid member on Patreon. These videos that I do are for free. So I have thousands of these kinds of videos out there. And it's through your support that allows me to do these things. So please help support by being a paid member on Patreon or by donating and going to my store, the-studio-reykjavik.com, and purchasing the products that I offer. I have a lot of new products. Again, the ubiquinol, the NAC plus, the NAD plus, the NAC, the ubiquinol, the K2, MK7, and the nattokinase. So get those from the store. Also get some of the other products that I have. The more that you purchase for your order, the cheaper the shipping will be, because the shipping I do is exclusive with FedEx, and I do it in a fixed rate for the shipping. So the more items that you purchase, the better you are with the shipping. So thank you for listening. Please go to my store, the-studio-reykjavik.com, and please ask your social media to follow me so I can build an audience and overcome the censorship and the throttling that is happening on some of these social platforms like YouTube. Thank you for listening and have a nice day.